Pronto?
No, pronto.
Confirma se eles estão estudando e vendo. Aqui já não dá para ver. Não pode. Pergunta se está estudando. Bruno, você está estudando? Can everybody hear us?
Tá tudo funcionando aqui, ó.
Beleza. Valeu.
Valeu.
Okay, so let's switch to English. Good afternoon, everyone. So today we're going to have Dr. Thiago Patente presenting his data that he's generating in Leiden University Medical Center at Holland, and he's going to talk about nutrient sensor mTOR and AMPK in the dendritic cells, the central regulators of tolerance and immunity. And Dr. Thiago Patente has his master's degree in cellular and molecular endocrinology by the medical school on the University of São Paulo, in São Paulo, and also PhD in immunology done here at the Department of Immunology with Dr. Alexandre Barbucci. So, welcome, Thiago. Thank you. All right.
So, good afternoon, everyone. Yeah, thank you for joining. Sorry for the technical issues we had for the lecture. But yeah, so I'd like to share a little bit of the data that I have generated here, partially here during my my internship that I was doing there, and now as a postdoc in the last few years. So it's about the role of mTOR and AMPK in the dendritic cells and how those nutrient metabolics, how they can coordinate metabolism and immune function of the dendritic cells. So a little bit of a background. So a little bit of the background. So the dendritic cells resides within the tissues in what we call immature states, so immature dendritic cells, and they can be activated and then become immunogenic dendritic cells upon immunogenic factors that activate those cells, and then they are prone to prime effector T cells. But on the other hand, they can also be exposed to tolerogenic factors, and then they can become tolerogenic dendritic cells, and they can prime regulatory T cells, for example. And recently, in the past few years, it has become quite clear the role of metabolism in priming and being very important for the function of the dendritic cells and other immune cells. And data from the literature has supports the idea that the activation of cells, like dendritic cells, they rely on what we call anabolic metabolism, where mTOR is the important protein in drive anabolic metabolism in different types of cells. And on the other hand, catabolic metabolism has been associated with the development of quiescent cells and tolerogenic dendritic cells, with fatty acid oxidation and oxalogs being important for this process. But we don't really know exactly how AMPK and mTOR control this process of driving anabolic metabolism, driving catabolic metabolism, how this is linked to the function of those dendritic cells. So I'm going to start talking primarily focusing on the mTOR complex 1. So the mTOR complex 1 is formed by mTOR and another protein that is its adapter that is called raptor. And mTOR1 is very involved in cell growth and proliferation. And you can see that AMPK is also linked to the mTOR function, though those proteins kind of counterbalance each other. And mTOR is central to both mTOR complex 1 and 2, with the difference of the adapter molecules like raptor for mTOR1 and Richter for mTOR2. So some data from the literature has already studied the whole of mTOR in dendritic cells, and they have shown, for example, that if you inhibit mTOR in dendritic cells, those dendritic cells are more prone to stimulate CD8 T cells response. Also, if you inhibit mTOR in those dendritic cells, you make those dendritic cells be more tolerogenic, and they promote allograft acceptancy. And also, a more in vivo data, that's like one of the few in vivo data with mTOR, is that if you lack mTOR in dendritic cells, you call them a disbalance in the lung CDC1 compartments, and this has an impaired antiviral immune response. So quite a few of these data were focused on in vitro generated human mold disease or in vitro bone marrow derived dendritic cells with field data on in vivo dendritic cells. So we started our project looking at mice with specific selective deletion of reper in CD11C expressing cells. So primarily dendritic cells, but of course other cell types also expressing CD11C. And we looked at the skin drain and lymph nodes of these mice. So immediately what we observed that had already been shown before is this dramatically reduction in the Langerhans cell compartment. So that gave us security that our mice were correct, that data has been shown. And also this increase in the cellularity of the migratory dendritic cells. So you could see that there was an increase in the number of dendritic cells, both CDC1s and CDC2s. And because of this overall increase in cellularity of this antigen presenting cell compartments, this reduced Langerhans cell frequency was not translated on number of Langerhans cells in these lymph nodes. So we looked at the surface markers and the phenotype of these dendritic cells, both at steady state and upon CPG simulation. And we observed a reduction in the MHC class 1 compartment in some of the co-stimulatory molecules, both in CDC1s and also in CDC2s, supporting the idea that mTOR is indeed important for this more immunogenic profile of those dendritic cells. And interestingly enough, those differences were not very mirrored in the Langerhans cell compartment. You can see that even upon activation or upon simulation with CPG, the Langerhans cells increase the expression of some of the co-stimulatory molecules. So because of that, we decided to better try to characterize this what was happening on these mice, and then we decided to simulate these mice with CPG and MHPV peptides. And then 7 days later, we harvest the lymph node of those cells to look better at the T cell compartment and the overall immune response of these mice. So what we actually observed was that despite the reduction in the MHC class 1 and some co-stimulatory molecules, these mice were actually better in clearing or in the CD8 T cell compartment of the cells. We observed higher frequency of tetramer specific or HPV specific CD8 T cells, and also the secretion of interferon gamma was increased in these knockout mice. So we were a bit surprised with that. We're not expecting that. And we decided to better try to characterize both the metabolism part of the cells and also the phenotype of the dendritic cells. So we developed what we call like Medflow, which was like a flow panel that had both metabolic proteins and also co-stimulatory and co-activation proteins. So we targeted some of the proteins that are enrolled in some key metabolic pathways. So GLUT1 for glycolysis, PKM as well, then ACC1 for fatty acids, synthesis, CB2A for fatty acid oxidation, SD8 cytochrome C for mitochondrial metabolism, amino acid uptake, lipid uptake, pentose phosphate pathway. So some of the key enzymes that we could kind of understand what was happening metabolically in these cells. And then we ran some unbiased analysis, and the first thing that we could observe is that we could nicely identify in the lymph node the Langerhans cell, the CDC1, the CDC2 dendritic cells on an unbiased way. And when we ran some phenograph analysis to identify different clusters, we could identify 13 different clusters within these dendritic cell compartments. And we could kind of immediately appreciate that some of the features that we had manually identified on a gating strategy were also correlated here on this unbiased analysis. So the first thing we could see is that between the knockout and the wild type, regardless if those mice were at steady state or if they were immunized with CPG, we could observe differences in this compartment that corresponds to the Langerhans cells. So it was reduced, as we also observed in the gating analysis. And so here is the expression of XCR1, CD172, and AMPK, showing that those are the Langerhans cells. So what actually caught our attention at first was that those clusters, they were differently modulated between the wild type and the knockouts. And we could see that those two clusters, clusters 6 and 10, they were here close to the Langerhans cells. And this cluster 6, it was a cluster that was upregulated in the knockout, whereas cluster 10 was downregulated in the knockout. So they were like, there was a shift in this Langerhans cells clusters. And when we better looked at the cells, we could see that they were corresponding to the Langerhans cells. And actually, what was interesting is that this cluster that was upregulated on the knockout, it was like increased PD-L1 expression, and also it was a cluster that had a bit more of catabolic metabolism, an anabolic metabolism with reduced ACC1, increased CPC1A. So there were like clusters that were differently modulated into these cells. So we tried to sort the Langerhans cells from these mice to see if those Langerhans cells could be driving this phenotype that we were observing of these mice responding better to the immunization as we were not expecting it. So we sorted the CDC1 and we sorted the Langerhans cells in mice that we immunized with CpG and OVA, and then 24 hours later we harvest the renal lymph node, set up a co-culture with the presence of OT1 peptides, and observed and looked at the proliferation of the T cells and the secretion of interferon gamma. And then, for our surprise, there was no differences in neither the proliferation of this CD8 T cell nor in the secretion of the interferon gamma by the CD8 T cells. Then we looked back and went back to our unbiased analysis, tried to look more carefully. And then from this analysis, one cluster that's, or one part of it that actually we initially had not paid much attention was this part here. So this CDC1 compartment that actually express high levels of EpCAM. So when we looked better at these cells, we could actually manually identify or on a biased analysis, by gating strategy, we could identify these cells that were double positive for EpCAM and XCR1, and we kind of saw that those cells, they were a bit more immunogenic than the EpCAM positive XCR1 cells. They were a bit more immunogenic than their counterparts that did not express XCR1. They were EpCAM negative. But because the number of cells that we could see here, that's also the expression of MHC class 1 was a bit higher in the cells, and they were not, the decreased expression of MHC class 1 in the cells were not as pronounced as in the EpCAM negative cells. So because of the scarceness of these cells, we were getting like 0.7% of the whole migratory cells. We couldn't sort the cells to try to see if they were the real responsible for it. But luckily, there was this paper from Nature Biotechnology that had proposed this alternative methodology where from the data, you do kind of a mild digestion of the lymph node, and you could preserve the in vivo interactions that were happening in the lymph node if you don't exclude your doublets from the analysis, and if you gate on the double positive cells for CD11C and TCR beta or CD2. So we run these experiments again, and we perform this mild digestion, and then what we could observe is that if we gated on this, using their gating strategy, so we didn't exclude the doublets and then removed the B cells, the PDCs, and then we looked at CD11C positive, CD3 positive, CD8 positive cells, we could observe that actually only these EpCAM positive, XCR1 positive cells, the frequency of this interaction, this interacting cells, it was increased in the knockout, but only in these EpCAM positive cells. So what we think is happening is that these cells are being important to drive this more immunogenic response that was happening on these mice. So we had this model where mTOR would be important in more specifically or in the CDC1 cells, where mTOR1 would be limiting the expression of MHC class 1 and CD80 and 86, and also limiting this the proliferation of the CD80 cells and the expression of interferon gamma cells, happening more in this CDC1 positive, EpCAM positive dendritic cells. So that would be like the first part of the talk, where we focused on mTOR1. And then when we looked at the other side of the balance at AMPK, that is upstream of mTOR and inhibits mTOR1 activation in a more direct way. But then we decided to focus this attention here to see if AMPK would be important to counterbalance mTOR and have like a more tolerogenic profile of the dendritic cells. So And AMPK is this small molecule that is composed of a few other subunits, and it is activated when you have increased ratio of AMP, so in energy starvation, and it promotes catabolic process and inhibits some other anabolic process in the cells. And some data that we had from a PhD student in the lab. So she was focused primarily in how AMPK was could be driving this tolerogenic profile of the dendritic cells. So she treated cells with 901, which is a compound that allosterically activates AMPK. And we measured the activation of AMPK by measuring the phosphorylation of ACC, which is the downstream targets of AMPK, the directly downstream targets of AMPK. And then she observed that when you treated cells with 901, there is an increase in the phospho-ACC levels. There is an increase in the IL-10 secretion by the T cells that were co-cultured with those dendritic cells. And also, if you harvest these primed dendritic cells, and then we co-culture with memory T cells from the same donor, we can see that those primed T cells, they suppress the proliferation of this bystander T cell, similar to what vitamin D does, which is a well-known tolerogenic compound in dendritic cells. So we had this model where where 901 activation, so increasing the activation would be promoting this IL-10 secreting Treg cells, and we questioned whether some tolerogenic compounds would also be activating AMPK in a similar way as promoting AMPK directly would be doing in dendritic cells. So we initially, we had a few compounds that we were using, but we narrowed down to retinoic acid, where we found like the more interesting results. So the first thing that we did, we looked at the surface markers of those of some markers in those monocyte-derived dendritic cells, and we observed that retinoic acid significantly affected the expression of CD80 and 83, so reduced CD80 and CD83, and increased a lot the expression of LAP, which is the surrogate for TGF-beta, CD103, and an inhibitory molecule called IL-23. Also, when we measured the activity of RALDH, which is an enzyme that converts vitamin A into retinoic acid and is known to be important to drive some of this tolerogenic functions of especially intestine dendritic cells, for example, retinoic acid promotes increase in the activity of this RALDH. And also similar to what 901 and vitamin does, if you do this suppression assay where we co-culture the T cells with the dendritic cells, harvest these T cells, co-culture these primed T cells with memory T cells, and see if these primed T cells are able to suppress the proliferation of these bystander T cells. So our primed DCs were able to also induce dendritic T cells that could suppress the proliferation of bystander T cells. And those T cells that were primed with these RA T cells, they also expressed a bit higher levels of FOXP3, and they secreted higher levels of IL-10 and reduced levels of interferon gamma. And we confirmed or checked whether RA treatment would induce the phosphorylation of AMPK or would induce AMPK similarly. And we saw that indeed, when we treat cells with RA, we see an increase in phospho-AMPK levels in these dendritic cells, and this is also associated with a reduction in phospho-S6, which is a direct downstream target of mTOR1. So you see this counterbalance of you have an increased AMPK and reduced mTOR1 in the cells. So in face of those results, what we decided to do was to further explore it, and we silenced cells for AMPK, and then we decided to see if some of these effects would be reduced. So the main effects that we were observing was that upon RA treatment, we had an increase in CD103, which is well established in the literature. There are quite a few papers showing that. And there was this increase in RALDH activity associated with an increase in the IL-10 production of these T cells. And when we silenced cells for AMPK, the expression of CD103 was at least partially reversed. Same thing happened with the RALDH activity, was also reduced, and the secretion of IL-10 was also at least partially reduced in this when we silenced cells for AMPK in these dendritic cells. We also were a bit further to see if that phenotype was also happening in mice. Then we generated bone marrow-derived dendritic cells from the CD11C AMPK alpha 1 knockout mice. And we observed a similar effect where the proliferation on the wild-type mice, when we treated those RA with those bone marrow-derived dendritic cells with RA, it was reduced the proliferation of these T cells, and then this effect was not present in the knockout mice. And also was an increase in the FOXP3, and that was not observed in the knockout mice anymore. This was all bone marrow-derived dendritic cells. Then, of course, AMPK is a kinase, so it's not a transcription factor. So we were wondering what could be downstream of AMPK, what AMPK could be doing in these cells to drive these phenotypes. And recently, there was this paper that came out where they observed, it was an in vivo paper, but they had this mechanism where AMPK and PGC1 alpha would be increasing raw H activity in the intestine of these mice. So we decided to look whether PGC1 alpha could be one of the mechanisms that was downstream of AMPK. We had some qPCR data from the cells, and we observed that both PGC1 alpha and PPAR gamma, which is also important in this axis, they were increased upon retinoic acid treatments, and then we used two inhibitors, one inhibitor specifically for PPAR gamma and another inhibitor for PGC1 alpha. And actually, what we observed was that while a PGC1 alpha seems to be important for, or at least partially important for the expression of CD103, PPAR gamma was not. But both the raw H activity and also the induction of these suppressive T cells, they were not affected by PPAR gamma nor PGC1 alpha. So most likely AMPK was not acting through PGC1 alpha or PPAR gamma to induce the suppressive effects. Then we went back to the literature, trying to find alternative pathways that could be happening in cells. And then another downstream target of AMPK was FOXO3, also a transcription factor that had been associated with AMPK for a while. And also FOXO3 was also associated with retinoic acid, was also linked, the treatment with retinoic acid had also been linked with activation of FOXO3 in some other cell lines. So we decided to give it a shot and see if that could be one of the targets. So we did some in silico analysis to see, for example, which site in FOXO3 could be a predictive site where AMPK could be acting, and that would be serine 413, which was a nice confirmation, in silico confirmation, because that had also been shown in the literature that this site is a site for AMPK. So our in silico analysis confirms that, and when we looked at some Western blot data for FOXO3, we saw that indeed this serine 413 was increased upon retinoic acid treatment, and that was associated with a reduction in the serine 253. And this dynamic is quite interesting because if you look at the literature, there are papers showing that, well, FOXO3, it's a protein that it translocates to the nucleus when the phosphorylation levels are low in some of the sites. And interesting enough for us, at least, this site is one of the sites that it is linked with the translocation of FOXO3 to the nucleus, whereas this site is not linked with the translocation to the nucleus. So it is just a site that is a surrogate for AMPK activation. So it suggests that AMPK, it is phosphorylating this site, and this would be associated with the translocation of this protein to the nucleus, where it would it would do its thing. Let's put it that way. So we silenced cells again for FOXO3 for AMPK. We looked back at the serine 413, and we nicely could see that it was increased. And then upon silencing of AMPK, this increase is not present anymore. And then when we silence cells for FOXO3 now to see if we could recapitulate this phenotype, CD103 does not seem to depend on FOXO3 expression, but the raw H activity was reduced when we silence cells for FOXO3, and the ability of these cells to induce suppressive T cells was also reversed when we silence cells for FOXO3. So suggesting that this axis AMPK FOXO3 is important for the retinoic acid signaling. Then, so far, what we had concluded was that retinoic acid would rely on AMPK for their tolerogenic properties, and that this induction of tolerogenic dendritic cells by RA was independent of PPAR gamma or PGC1 alpha, but would be dependent on FOXO3. So because RA is also really, really present in the intestine of these mice, and there were quite a few data showing dendritic cells are important in the intestine to keep tolerance and to induce regulatory T cells there, we decided to take a look at the intestine of these mice that were knocked out for AMPK. So in the intestine, we have primarily three types of dendritic cells, we have the CDC1 and the CDC2s, but the CDC2s are divided in the CD103 positive dendritic cells and the CD11b positive, CD103 negative dendritic cells. And the CD103 single positive, they are the CDC1s, and both CDC1s, CD103 positive and CDC2, CD103 positive, they have been associated with the development of regulatory T cells in the mesenteric lymph node and also in the intestine itself. So we started by looking at the AMPK signaling in different tissues of these mice, and interestingly enough, the AMPK signaling seemed to be higher when we looked at dendritic cells that are in both the colon and in the small intestine, and even more specific in the CDC2s that do express CD103, where we over here. And this AMPK signaling was also associated with an increased broad H activity in these dendritic cells. So you can see that the dendritic cells that do express the double positive dendritic cells, so the CDC2s expressing CD103, they have the highest level of phospholipid C and also the highest levels of broad H activity. This is in the small intestine of these mice. And the levels of both AMPK signaling and broad H activity are way higher when you compare to macrophages, T cells, or B cells. So we again performed some unbiased analysis in these mice, and we could nicely identify CD103 single positive, double positive, and CD11b positive cells in these mice. Again, a few clusters identified, but two clusters were actually quite interesting that popped up on this analysis. So a cluster that was upregulated in the knockout mice and another cluster that was downregulated in the knockout mice. And these clusters were actually interesting because the cluster that was downregulated in the knockout mice is actually a cluster with high broad H activity and low expression of CD86 and CCR7, whereas the opposite is on the wild type mice. So that kind of suggested us that these knockout mice, that when you don't have AMPK in these dendritic cells in the intestine, these dendritic cells are a bit less prone to induce regulatory T cells, and they are a bit more on an immunogenic profile. So we decided to test if those dendritic cells, if this effect that we were observing there, how that could be translated in a disease phenotype, if the disease phenotype would be enhanced in these mice. So we are over there in the parasitology department, not in the immunology department. So we have sometimes to use some of the parasites that we have there to justify the fact that we are there. So we have access to Schistosoma mentionii and some other parasites. And we then infected these mice with the Cercaria parasitic form in the mice. So the schistosoma, it has like a life cycle that is quite complex, but upon in humans, you have the infection with the Cercaria. They migrate through the lungs, and then they reside in the liver. In the liver, they mate, they shed eggs, and then they migrate, actually they mate, they migrate to the mesenteric vessels, and then they establish themselves in the mesenteric vessels. They shed eggs there, and these eggs can break the intestinal wall. Part of these eggs are eliminated by the host, but part of these eggs goes back to the liver and they lodge in the liver. And these eggs are important in the Th2 associated response that we have in the liver, like the egg granuloma formation, for example. But through the course of the disease, what we do have is that in longer times, like around 16 weeks, for example, the Treg response seems to be important to control this egg granuloma that we have in the liver. So because we were looking in the Treg response, we infected the mice and we sacrificed them 16 weeks after the infection to see if the regulatory response would be affected in these mice. So when we looked at the liver of these mice, we actually observed that indeed both frequency and cell numbers of regulatory T cells were compromised in the infected mice that were that did not express AMPK in their dendritic cells. So there were reduced phospholipid frequency on numbers, and that was associated with an increase in the Th1 and Th2 immune or cytokines in the hepatic lymph nodes of these mice. And also, when we look at the size of the agranuloma that we observed in this mice, there was actually an increase in the agranuloma size of these mice, suggesting that AMPK expression in dendritic cells is important for these dendritic cells to induce regulatory T cells and to shut down or to prevent the increase in the agranuloma side of these mice. So next, we also used an acute colitis model to see if a similar phenotype could be observed in these mice. And we indeed observed that mice that were knockouts for AMPK in their dendritic cells and were exposed to a 2% ESS over the course of 2 days, they lost more weight than the wild-type counterparts. That was also associated with an increase in the colon length and colon weight, an increase in the inflammation score, and again, a decrease in the ESS in the CD103 double positive dendritic cells, with now an increase in the single positive dendritic cells. But this time, I forgot to put the image here, but the Treg compartment was not affected in these mice. So both the Treg were completely equal in all of these mice. However, what we did observe was an increase in the Th17 cells. So we observed an increase both in the frequency of HORIMT positive, FOXP3 negative T cells in these mice, and also an increase in the IL-17 secretion of these mice, which kind of is building up the next steps in the lab on trying to understand how AMPK could be in the intestine controlling this balance between Treg and Th17 response. So what we had was like in one side, in one tissue, like the skin, and in the CDC ones, mTOR would be important to prevent this overactivation of the cells. And then when we switched side and went to the intestine in the CDC2s, for example, the AMPK was important to drive Treg response and to prevent overinflammation in the Th17 model. And then what we were still trying to identify is, for example, in the intestine, which factors could be promoting this activation of AMPK. And we have some candidates like some short-chain fatty acids, like butyrate, for example, which is quite well established tolerogenic compound as well. So we are trying to identify what could be driven this AMPK, increase AMPK signaling in these dendritic cells. And of course, this whole dynamic of AMPK and mTOR being counterbalanced, how exactly this is happening, because not always it's black and white where you have an increased AMPK or reduced mTOR or vice versa. Sometimes both of them go up at the same time. Sometimes both of them go down at the same time. So it's still not black and white situation. And yeah, with that, I'd like to thank everyone in our department that helped us draw this data, and also you guys for the time and to allowing me to share some of the data here. And I'm glad to take any questions that you might have.
Can everybody hear us at Rio as well? No? Are they hearing us?
I hope so.
Turn on the mic. Anyway, I'd like to open for questions. So first, the students have any questions, either here or from Rio, please. If I would like to ask them.
Can I just ask a quick question, or just to warm up?
Yeah, sure.
The audience. There's actually two technical things, so very specific. One regards your doublets. So you this technique.
Yeah.
Why you open up the gates for doublets instead of just making another gate to specifically get doublets? Is there any specific reason for that?
No, we actually, the only reason why we opened it up entirely was just to follow up the same gating strategy that Nature Biotech papers actually did. But when I was doing the analysis, I kind of did both ways, and the result did not change significantly. So you could see differences if you get only the doublets or everything, kind of small differences in frequencies. But the overall response was still the same. It was the same profile that was having there. So it was more like to just follow up the same.
The seamless did not contaminate.
No.
Your
Yeah.
And the other thing is that the T cell suppression assay, when you first show, I had the impression that you were talking about the CD8s, but then you mentioned a lot CD4s and IL-10. So just to clear up.
Yeah.
So,
You mean here. So yeah, the part of the AMPK that was all CD4.
okay, everything was CD4.
The part of the AMPK, everything was done on CD4 T cells. So they were all human. It was all human data. Like here, for example, there was all human data. Here, yeah. So this is all human data. So we have. So what we do, we have Buffy codes that are quite kind of like the peon we have here. And then we isolate CD4 T cells from one donor. We isolate naive and memory, freeze them down, and then we just thaw it when we need to use it.
It's the regular T, right?
Yeah.
It's
Yeah.
No CD8.
No CD8.
That's a P.
Yeah. No CD8, no good one.
All right, so we have another question. Let's open up for maybe your people. No?
I think, wait, there's someone raised a hand. Yes.
Yeah, I actually did.
Okay.
Hello, Thiago. Thank you very much for your talk. It was very, very interesting. I'm not sure if I missed something, but in your S-Masonite model, you showed only data on 16 weeks, and was wondering if your genetic maneuver to delete AMPK also affect skin dendritic cells and affect these earlier phases of the response. How is the number of eggs in the feces of these animals, or even adult worms? Are they changed?
Good question. So for this experiment we didn't have the egg counts of this mice in this experiment, because I think it was like a very kind of an oldish experiment that we managed to kind of save it after a while. But what we did, we had also data from 8 weeks of infection, and 8 weeks of infection we had a very, very similar response, very similar result. So we also observed the same effect in the regulatory compartment in the liver of these mice. The only thing that at 8 weeks we still were not able to see differences was in the egg granulomocytes. So the egg granulomocytes, the differences there only appears at 16 weeks, but not at 8 weeks. And we also tried to get data from the intestine of these mice on both 8 and 16 weeks, but it's very, very tricky, and the tissue is very fibrotic, and it's very hard. So both of the times, a few times that we tried to do the experiments, we did not manage to get cells from the lamina propria. So that's why we kind of focused a bit on the lymph node. I didn't show the lymph node data because there was not too much thing going on there. But the phenotype seems to be a bit more restricted to the liver.
Thank you. I don't think we have any more questions from our side here for now.
I have a quick question, but it's also following what you said about AMPK in the skin.
Yeah, good. Yeah, so that's actually it's on the pipeline to be done. Like now that we have the licensing again, because we haven't looked at the skin. We've tried a few protocols to get the skin, the skin from the dendritic cell, the dendritic cells from the skin itself for the first paper. But the few times that we tried, none of them we actually were able to get a robust number of a good number of cells. So that's why we kind of focus on the skin draining lymph nodes, because that's
And
at
then
least we
the
could get.
immunization CDH. Yeah,
Exactly.
because I was just, what are your thoughts about AMPK and tolerogenic dendritic cells in the skin and some type of tolerance to skin microbiota?
Yeah.
Things like that, what that could mediate tolerance for not generating any kind of autoimmunity in the skin, for example. It's named with toll-like receptor 7. Agonist.
Oh, yeah. Psoriasis, yes. Yeah, so what I've noticed with AMPK is that it's not a black and white relationship as we thought it would be. Because we started off that thinking that AMPK could be some sort of central mechanism in driving photogenesis of dendritic cells, and that kind of different types of agonists could be acting through AMPK. But we immediately realized that that was not the case, because we've tried vitamin D, we've tried retinoic acid, and we've tried dexamethasone, and dexamethasone does not act through AMPK at all. Doesn't do anything. Vitamin D is a tricky and interesting situation, and well, we find a lot of levels of vitamin D. So I think that it could be that AMPK plays a kind of a role in the skin dendritic cells, but maybe a bit less than in the intestine, because vitamin D, it does, what we observed was that vitamin D, it does increase AMPK signaling, but AMPK signaling is not important for the vitamin D driving properties. So if we silence cells for AMPK, or if we look at the knockout mice for AMPK and treat the cells, both the bone marrow derived dendritic cells or the mold disease with vitamin D, in the absence of AMPK, they keep on doing whatever they do. I mean, they metabolically they are affected. So all the metabolic changes that we observed in the vitamin D treated dendritic cell, that was reversed when we knocked down AMPK, but they were still able to induce functional or regulatory T cells. But in case of psoriasis, which is more like a Th17, where we do have the Th17 balancing there, so that could be interesting, because we also have some in vitro data showing that when we silence cells for AMPK and then we treated them with zymosan, for example, the Th17 response that is induced by zymosan is hampered. So it seems that indeed AMPK kind of, depending on the tissue or the microenvironment and whatever is like surrounding the AMPK, can kind of control the priming of, or the generation, let's put it that way, of a Th17 or a Treg priming a T cell or DC. So it could be, it's definitely one of the things that we are now in our new ethical committee thing where we're trying to look at.
Gustavo.
Yeah.
Yeah, no, it's just that I don't know if you guys noticed, but we have a question in the chat from Vinicius Borges.
No, I have a question. What are the main differences between the dendritic cells and macrophages in immunometabolism in concern of the role of mTOR in driving catabolism and activation? That's a good question. Well, personally, I haven't looked much at the mTOR signaling in macrophages, but what we do know is that mTOR is also important to drive this so-called M1 macrophages. And in case of the dendritic cell activation, it's I think it is still very unclear, because most of the data also focusing on macrophages, or quite a few data focusing on macrophages, are also in vitro generated. And when you look at the dendritic cells in vitro, also, it's pretty kind of black and white. If you just like inhibit mTOR, you have these more tolerogenic dendritic cells, so everything fits. But when we go to the in vivo settings, as we showed here, it's not super black and white. So we have mTOR that is kind of driving this catabolic metabolism, but instead, in one type of cells, it is it is blocking the immunogenicity, but in other cell types, kind of driving more immunogenicity. So it's kind of the short answer is I really don't know, but we have to kind of look better there, because it's not as black and white as we thought it was. That is for sure.
Okay. So any further questions? Yes. Everybody shy? No?
May have one.
Yeah.
Did you type amino acid transporters and metabolism in the living cells?
We checked CD98 expression in both in vivo and also in in vitro generated, but there was at least in the surface marker expression there was nothing actually there. But it is interesting is that let's see. We also did some metabolomics analysis in those RA-treated dendritic cells, and one of the few things, also not many things popped up there, but one of the few things that actually popped up was kind of overall reduction of some amino acids, like methionine, serine, some of those nitrogen metabolisms overall, that was indeed reduced in the metabolomics of the cells, but it was nothing super black and white. Surprisingly enough, at least for us, looks like RA does not act through much via metabolic changes in the cells to do its thing.
Any other questions? No? In Rio, Bruno?
No, we don't have further questions here.
Okay,
All right, then.
so thank you very much, Thiago.
Thank you.
Thank you. It was really great talk.
Não, amanhã eu fico não posso, porque eu estou todo dia agora. E aí, você está aqui por quanto tempo?
3 semanas.
Umas férias?
É.